Okay, so like I said, the most important thing is you begin with the properties of lipids. And in that, what are the different types of lipids? And, translating that chemistry knowledge into actual properties of how you see them, how you see and how they behave biologically in terms of like how they're solid or they're liquid and how, that takes place. Okay, the second important thing. So I said the first important thing was that learning about the properties and what is the saturated versus basic definitions and properties and saturated versus unsaturated. The second important thing would be nomenclature. I was very excited to see that most of you, whatever I checked yesterday and day before, that nomenclature was something that I feared that many people would not get it right, because sometimes people have that problem. But whatever I checked, I think I saw a pretty good success rate in terms of recognizing what is what. So great job on that. Just it's very simple. As you see, you're counting the number of carbons, counting the number of double bonds, and in delta, you place the position of double bonds. And that is what it is. And if you see here, that is the same thing, just this is a more complex. So you can expect this kind of question in exam. You are expected to know this. And it is also possible that there are many different questions in the question bank. So you probably will not have, you will probably waste time if you were going to look for something in PowerPoint. It's unlikely to be here. It's just something very simple if you just know how to do it rather than look for answers, because it's much quicker. Count the number of carbons, count the number of double bonds, put their positions, and then from the right-hand side or the methyl end, you can put the omega by saying, where is the first double bond? I would like to know if you have any questions about this, because this is something which is important, of course, for learning and exam, both purposes. So is there anything that you want to learn about this one? Okay. Okay, so now we get into some of the other parts. So the first part would be, of course, the biological implications, which is, which one is good, which one is bad, and what happens when you take saturated diet, which increases and which does not. So that's important. How does that translate into a person having actually just high cholesterol or increased amount of bad cholesterol, and that results in the clogging of arteries. Also important to know, just this one slide, separate slide, and this is on the essential fatty acids, and those are the omega-3 and omega-6, which are really needed for proper functioning of the brain. And however, you are not able to synthesize them, so you need to have them externally. You need to make sure that you are consuming them. There's just the list. It's not something I'm ever going to ask you that, okay, what is the other name of stearic acid, or expect you to remember any parts of this table. Functions, of course, you probably already know some. It is not difficult to imagine that if you are deficient of fat, then you might have dry, scaly, or rashy kind of skin, rashes of the skin. Also, it creates cognitive problem, because we talked about how omega-3 and omega-6 are important for brain development. And that always, kind of reminds me of that enriched like milk that you get. It says like, and it's actually much higher price, organic and DHA, omega-3, all of that, those kind of supplemented food sources are available. Okay, so I would say that I would still include this in the first part, because even though it is a lot, but this is one big chunk, the chunk of triacylglycerol, where you are actually looking at an example of combining a fatty acid with an alcohol and making that glycerol. And remember, these chains can be different, and that differences can give it different properties. In this case, you see this chain is exact same length, exact same thing. That may not always be true. Okay, we have talked about this in carbohydrates. So I would say this is a great place where you will be able to compare your knowledges and combine them and say, oh yes, I do know that when a glycerol and fatty acid combines, there is a condensation reaction, which means the water comes out. So that is to keep in mind. And also there is a hydrolysis in which it can be broken. So this is simple and mixed. I told you that they can have different chain. Okay, I'm just going to move forward because these are some of the things that you can do on your own. It's just the different kinds of properties of these materials. And here comes the difference. What are the differences between them? So at least this is a part of this module. So you're expected to know, okay, how is this different from the carbohydrate? I would say that these three reactions would be like the first part, or the end of the first part, where you are talking about different reactions. So one of them is saponification. The saponification is literally the formation of soap. And that would be something we'll describe in 2 slides from now, which is right here, actually. Saponification, where you're combining with either a sodium or a potassium hydroxide to make soap. And if you see here, this is what the structure looks like. And that's what, your bar soaps are like that. All the shaving soaps are like that. That's what all the soaps are. So important application. Then you have hydrolysis and hydrogenation. Hydrolysis is like opposite of condensation. You're using water to break, and condensation, where they're coming together, the water is coming out. So you have to remember that part. Hydrogenation. I think there was this question in assignment 3, which I'm happy that I'm sharing with you some of the response. I think some people got it and others did not. So hydrogenation can only happen if there is a double bond. So the question is, do waxes have double bonds? So let's answer that. I know there is no structure in here. Let's see the structure. I personally don't see any double bond. Please don't count this as a double bond. I have told you many times, this doesn't really count as a double bond unless it's between the two C's. I don't see any double bond. And you may say, maybe it's there, you don't see it. No, if there is a double bond, they won't never write it like this. Okay. It needs to be broken up. A CH2 is not going to be there. It's going to be CH. So as long as you don't see any double bond, how are you going to be able to add a hydrogen? You're not going to add hydrogen. Hydrogen can be added only if there was a double bond. You can see here the transformation of CH double bond CH to CH2 to CH2 dash CH2. And so that is possible only in that. So don't get fooled by this. If you know the fundamentals, you will be good. You know that if you're given a molecule and said, hey, can this undergo hydrogenation? Just look, no double bond. Can you provide an answer with explanation? Always provide. I have seen a lot of people will just not even read the question. When it says that, you say, yes. And the reason is it does have 2 double bonds. So clearly mention the reasoning, and that really helps understand what you're doing. And remember, in any exam, I have always seen there are people who will try to write literally as minimum as possible. If it conveys the meaning, that's fine. But then there are people who would clearly take the time and write down the explanation. Even in this case, someone would write down, yes, there is a double bond in position, I don't know, 6 and 10. And those can be hydrogenated. So someone who takes time and write, you can clearly see that they have a better understanding. So I would hope that you would take that, if there's 70 minutes, you would take that time and finish it off in a proper manner. So this kind of ends the, with waxes, it kind of ends the long first part. And I would say that would be, if you are doing that, you finish that and you like literally, I would say at least take a break of some time. Is it like having dinner or having a cup of coffee or going for a walk or whatever? Don't try to put all of that in one go. Okay, so then the next part, and it does clearly have a break in here, right? So the next part is the structural part. And the reason I said separate it out is because the structural part has a lot of names. So if you want to start this, you start this with a completely fresh mind. You're going to look at each one of this, learn about each one of this, what is sphingolipids, what is, glycolipids, what is phospholipids, and we are not going to deal with this, but it's It's also a part of this. We are not going to work on archibacterial atherolipids. So in this one comes lipids and what are their applications. So what is present in your cell and what do they do in the cell? So we're not talking of a totality. We're not talking about organs. We're not talking about arteries or plant BVAX or how the vaccine coating helps. We are talking about the molecular or the cellular parts of the lipid and their functions here. So that is to be kept in mind. Okay. So in this one, even though I will not be going through each and everything because I don't want it to be a super long session, I want to have time for you to ask me any kind of questions that you have in mind. So definitely you need to find a way to do it. Like I said, what I typically do is I come up with things that help me understand, even though it may be funny, crazy, or it may make no sense to others, it doesn't really matter. So when you are doing this phosphoglyceride, you have to remind yourself, phospho means I will have a phosphate group. Glyceride, okay, so I know that it has the glycerol part and the fatty acid. Ide is for the fatty acid part. So that way I know how a phosphoglyceride, I'm able to break it up in my mind and do it. Similarly for this one, sphingomyelin, and this is for myelin sheath, that's why it's sphingomyelin, but it's sphingosine, and that is why it does have this different kind of name. So find a way to do it. There are different ways. I remember making sometimes, those figures and cutting them out and keeping them next to where I'm studying. And that's how I would remember them, or make good notes, which you can, look and whenever you are thinking about it. So phospholipids, glycolipids, all of those, all the three things. Glyco. Guys, whenever you think of glyco, you should immediately turn your attention to carbohydrates. Glyco means there has to be a carbohydrate component. So there was this thing in oral exam, and as a lot of people, I was a little bit surprised. The question was about what is let me see if you guys can answer, what is the bond that connects two monomers in a carbohydrate? I don't know if you can answer that now, if you remember that, but you should be knowing that. You should be knowing what can connect two bonds in, what is the connecting bond in the carbohydrates called? It's something to do with glyco. And then what is glycogen? That is also related. So some of these keywords should just help you in memorizing that. So again, this is just the structural description of each one of this, which was on the bottom here, shown in small on the top, and just each of them. Then this particular part and the couple of slides from here, definition, examples, function, definitions, examples, functions, and that's how it goes on. At least you should remember the basics of it. Like you may not be, like, for example, yes, that is correct. You need to. So for example, I would not ask you, can you draw a glycerophospholipid with a myo-inositol group? I would not ask you that kind of question, but at the same time, I would expect you to at least know that glycerophospholipids has how many parts to it. What are the different parts, or what are the different things that comprise or make up a glycerophospholipid? The name itself is self-explanatory here. So learn to break it down instead of thinking, well, that's a long name, I'm never going to be able to say it. Try to say it, try to embrace it. Okay. And what are the function? It provides the function in here, and we have, I think there is more discussion of it later. Sphingomyelin, not hard to convey that this is a part of myelin sheath of nerve cells. And then the glycolipids and their roles in distinguishing different types of or type of the blood, or blood typing, or different types of blood cells, RBCs. Steroids, again, what are the different kinds of steroids? This is also going to be covered in later modules. So the quicker you learn to be like, okay, I'm going to take my time, sit down and think, okay, there is 4 different types of steroids that I was taught: cholesterol, estradiol, testosterone, and cholecalciferol. So all of these are important. And let me take a look at their structure. Oh, these are mainly fused like ring, like I guess ring structures made up of 3 6-membered and 1 5-membered ring. And you can see that commonality, at least in these two, actually in these 3, and cholecalciferol or vitamin D is different. And their functions. So this second part is knowing the structures, knowing individual, and knowing their applications in cell membrane and contribution to the cell, even cell identification. What is the fluid mosaic model? How things will pass through it. There is a passive transport means across the concentration gradient, and active transport means actively transporting that. Okay, so that is the second part. And then the third part, which is much smaller. If you wish, you can do it with the second part if you're comfortable. But it's a lot of random lipids. The first one is more organized. The second one is a little bit complex structure-wise. Third one is very random. That's why I said, again, you could take a little break and come back and try to learn this, where you are looking at all of these different lipids and their functions. Okay. So I would like to pause here and ask, we are right at the 3 o'clock mark. Like, is there any questions that you have from module 3 for me? If not, then I'm going to stop sharing and I'm going to start working on pulling out module 4 for you. Also, please remember that my offer always stands in the sense that if you have any questions for me or a feedback for me overall, this can be changed or this can be done, I would rather you say now so that we can make it better for you for the rest of the semester, rather than, of course, you are welcome to provide a feedback in the end, but that will benefit the next set of people. And I would like it to benefit you if it is something that can be changed. I do also want to mention one more thing before we start module 2. So I want to know if it is beneficial for you if Simone could stay back 30 minutes after the class and you had something individual that you would like to ask her after or before the class. Is that something that any one of you would like to use for a certain purpose if you cannot come on a Wednesday or a Thursday? Does it benefit you if you start early and someone is there to solve only your, let's say, specific assignment or concept that you were having trouble in understanding? If so, you can please let us know, because I would like to provide you more support for the next half of the semester. And I always do that. I'll always try to make sure that you're more supported the next half. And Simone volunteered by herself. So thank you so much, Simone. I really appreciate that. And so if you have something you can ask, if you would like to stay back today, I'm pretty sure she can stay back and answer some questions that you might have specifically. Okay, so now for the next part, that is the module 4. So module 4, I feel like it's something that is very, it can still be divided in different parts, but I feel like, I personally feel it's way more manageable than module 3 because there is less to memorize and a lot more of logical things. The only thing is that if you have never done a DNA structure before, you may find a lot of new terminology. And so that may be the only challenge depending on if you've done it or not. So with the basic definition comes those different parts, which is the purine or pyrimidine base, pentose sugar, and phosphate. And I expect you to, again, be able to compare it with different things. For example, compare it with, like I said, carbohydrates, because if I ask you I'm not saying this is a question in the exam, but if I ask you, what is the sugar present in the nucleotide? You shouldn't be shocked. You shouldn't be like, there's a sugar in the nucleotide? I didn't know that. You should be immediately able to identify that, yes, I'm talking about pentose, which is a monosaccharide, which is a kind of sugar in the DNA. And the reason why I'm saying is sometimes these are the kind of responses I would get in the oral exam. I think a lot of people did pretty well, but if you were confused, I just want to come to that and make sure that is known and clear to you. So if you see here, the definitions of nucleotide, nucleoside, and nucleobase are shown here. The most important thing for you to know is what are the different parts of nucleic acid? How do they make up the DNA? So the first one of them is the sugar, and this slide is dedicated to make you understand that DNA and RNA are different just because they're different, they have different kind of sugar, and they have one of the bases that is different. A thymine in DNA is not present in RNA, and what is present is a uracil. So the definitions, and I personally would expect you to know these three parts really well. So well that if I show this to you and I tell you, I'm not expecting you to tell whether this is adenine or guanine, that if I show you anywhere, you can at least tell whether it is a DNA or RNA, and you're at least able to label the three parts, that is phosphate, deoxyribose or ribose, sugar, and the base. So you are able to make that distinction. We're able to recognize and make that distinguish. Okay, so there is purines and pyrimidines. And like I said, the way I personally remember pyrimidines is by noting that pyrimidine has a Y, cytosine has a Y, thymine has a Y. So everything that is Y is pyrimidine. Whereas in case of purine, that's not really true. These don't have Y, and this doesn't have Y either. The only thing that I must remember always is that that's not for DNA, and that would be that T is replaced by a U in RNA. Sorry, this is going to be crazy, writing it like that. So that is the only difference between the bases, but otherwise, there is a lot of similarities in the structure too. Expect you to know completely the double-stranded, the structure of DNA. I'm not thinking there is a detailed question on that, but I think there is a lot of value in knowing that there's a 5' end and a 3' end, and that 3' and 5' are inverted here. I hope you know that. Whenever I explain this, I do feel like some people get lost here and it's not easy to remember. So the way to do it is you start naming the carbohydrate right from the anomeric carbon. You all know what is an anomeric carbon. So right from that position, it doesn't have to be anomeric carbon, but that position. One, in this case, it's not anomeric because it's actually attached to nitrogen, but you start here, 1, 2, 3, 4, 5. And so this is the 5' end, and then this is 3, so that is 3'. The presence of double and triple bond. Also for you to remember, I'm going to write it down here. A pairs with T and G pairs with C. And that particular rule is kind of broken in RNA. So in RNA, A complements U. It's not pairing. So I'm trying to just show you a complementary base pair is U. Actually go back. Thought it went back. Okay. So the structure of RNA is different from DNA because there is only one strand. And now you may say, why is there only one strand? Remember that ribose, yes, that difference between ribose and deoxyribose is enough to change the structure of RNA. It is not the uracil, it's the main structure of the sugar that causes this change. Okay, so the first part, like all understanding what exactly it looks like at a very molecular level, what are the different, in terms of chemistry, how does it work out? And now we start looking at what does it do in the human cell. So for that, we have to know that the DNA, the role of DNA is to make protein, and it can do that only via the RNA. It cannot do it otherwise, right? It cannot just make a protein. So just the definitions of haploid and diploid and what it is like. Let me ask you a question and see if people remember and if people are alert and listening. So let me just pose you a question. Okay. So my question is, a dog has 78 chromosomes. Okay. So we have 46 chromosomes. A dog has 78. What is the haploid number of dog? That is my question to you. So any dog lovers here? Dog has 78 or 78 chromosomes, which is 39 pairs of chromosome. The answer is actually within the question itself. What is the number? What is the haploid number of chromosomes in a dog? Just waiting for if there is anyone else who has anything else to say. I'm only going to say if I see like at least a couple more responses. So again, let me just restate here. And my question here is that I'm not using the word, I'm not using the word diploid or haploid. All I'm saying is a dog has 39 pairs or 78 chromosomes. And what is its haploid number? And you know, it's kind of crazy to know that they have way more chromosome than what we do have. Okay. So yes, I did hear from 3 people, 39. 39 is the right number. 39 is absolutely the correct number. Okay, so now I'm going to reverse this question and I will say, for now, you know, we should be making the cat lovers happy too. Cat has 19 pairs of chromosomes. And now you do know my question, and that will be, what is the diploid number? Okay, so what is the diploid number? That's all the simple question here is. Okay, I see there are some responses in here. Yeah. Yeah, so this is, there's definitely the right answer, 38. So please note that when we're saying 19 pairs, that 19 pairs, that 19 is haploid and the times 2 is the diploid number. Okay, so just, it's good to know some of these. So if you are interested in this kind of thing, you know, the chromosomes is always like, it's always a very interesting topic to learn about. I'll give you one last example, and that would be trying to see if it's showing the diploid. Give me one moment. I'm trying to figure out if it is giving me 46 various human being. Okay. Yes. Okay. So similarly, if I say that the chromosome number, and I'm going to tell a couple of organisms here, like, you know, 48 chromosomes are present in chimpanzee. There are 48 chromosomes in orangutan. There are 48 chromosomes in potato. And there are 48 chromosomes in a particular kind of, in a hare, like a rabbit, right? So if there are 48 chromosomes total, then the haploid is going to be 48 by 2, that is 24. And like I said, just it's a good moment to remind you that number of chromosomes have nothing to do with exact adaptability, because there are lots of repetition, lots and lots of repetition in chromosome. Our every bit of DNA is not being utilized towards creating something in the cell or making us smarter. Okay, that's not exactly how it is. Okay, so what are the different types of RNA? You are not going to be going into details just because it's an introductory biochemistry class. So what are different types of RNA? What is the comparison between DNA and RNA? 100%. If I would be you, I would pay a lot of attention on how is DNA and RNA different? What is the relation between DNA and RNA? And we are going to explore it by doing a problem, which is the kind of thing that I expect you to know 100% for the exam. And that is this one. So as you know, the definition of transcription and translation, transcription is DNA to RNA and translation is RNA to protein. And the easy way to remember is that DNA to RNA transcription has a C in it, and this is translation has an L in it. So C comes before L. So this transcription comes before translation. A lot of times in exam, I would ask, not just your class, in a biotech class or in a biology class, I would ask the process of conversion of DNA to RNA. And then there are two options given, transcription or translation. So if you write the wrong answer, you're going to really, really hurt my feelings, okay? So this is something I'm very, very particular that students learn properly. And I always, make a note of it that people don't know what is the difference between transcription. So please know that DNA to RNA is transcription and RNA to protein or polypeptide in this case. Polypeptide is same as protein. Its poly is many and peptide indicates the, amino acid. So polypeptide, and that is the process of translation. So let us pause for a minute and see how this is happening, okay? So if you see here, at this time, I would like you to just focus on the top. So I'm going to cover this, okay? So that you're not, attention is not getting diverted. And I would like you to remember that A pairs with T and G pairs with C. In case of RNA, we will, DNA, in case of DNA, we will come to RNA when we come to RNA. DNA is to be replicated. When the DNA is to be replicated, the DNA opens up and there is single strand. What is the nucleotide that will complement this? So you have to remember that T pairs, opposite of T is A and opposite of A is T. So if I would be you, I would write down this rule on the side of my rough, like a worksheet that I need for workout for these problems. And so then I'll keep going. TAAT, CG, TA, TA, and then G. This is something that will not be provided in the exam. You are expected to know this. You are expected to have this very simple pairing rule memorized. Yes, thank you so much, Kayla. I really appreciate you doing this quickly. So this is great. You can see that will be matching. We will be coming to that. Now, what happens in case of RNA? Like if you are smart, you would have realized that T is just replaced by U. So this is still A. However, the RNA does not have a T. So it's U. And then this is G, same. This is A. It doesn't change. It doesn't change, nothing, no change, except for T is replaced by a U. Sorry, this is like not always easy to remember. Okay, so if you see here, please note the difference between them is T replaced by a U. So when DNA is replicating, this is what the strand looks like, or this is the DNA strand looks like that. And when it's replicating, also it is putting new nucleotide, that's what's going to be. However, when the DNA is transcribing, and I'm using the word transcribing purposely to indicate transcription, then it is going to be U replacing T. I want to pause here and ask you, are there any questions? You will be expected to know this in the exam. Whether it's this exam or final exam, you're expected to know how to do this. So I'm just going to write here the rule, if you will, in red, just to indicate that we are talking about RNA. So A pairs with U and G pairs with C. And that's what makes it a little bit, different. So are there any questions at all in here? Yeah, there's no question. Is hoping people have understood this. I saw something really, really funny last year. It's like unbelievably, I was like, wow, people did, or maybe I never conveyed the meaning properly. And the unbelievably funny thing was that while making you practice, let me just go to that slide. I had these questions for you, right? I'll show you. I had these questions for you. So when I gave a similar question in the exam, or even in the assignment, people just did 2 and they left. No. I was just showing you how to do it. You're supposed to do the whole thing. Why are you going to stop at 2? Because I'm not going to be the one working out the rest of it. So this typically carries a good amount of points. Good news is it's very easy. You can convert the DNA to RNA, and you can convert the RNA to protein. And for that, let me just remind you, you take the chunks of 3 nucleotide, which is called a codon, and it codes. It's called a codon because it codes for an amino acid. So for in this case, it's A. Oops, that's not the right color. So A, and then U. And if you're smart enough and you see it already here, no problem. If you feel like you just want to go a little bit more like this, so AUG will be like this. Oh, yes. Good question. Good question. So 5, what does it mean in the front? I will actually, thank you.
That is a good question. I was going to ask that. Thank you.
Yes. Yeah. So good. This is what I really appreciate, because, if you don't ask, then we will not be able to review this kind of thing. So CUA, so C, and then U. And so this is your CUA, which is a leucine. And so you are expected to go about completing this entire thing. Please do not leave it for me. If it is a 5-point question and you did only 2, you will get 1 out of 5. That's all you will get. Okay. So now let us go back. So is there anything that I can help you clear about how to transcribe and how to translate it? And I would like you to use those terms instead of using the term convert DNA to RNA. And even if we use that in the question, when you are saying it, transcribe, translate. Okay. So are there anything, is this table confusing? I know this part may be confusing because I think some people wrote stop codon as W, but stop codon is, there are 3 stop codons. Can't even see the rest. It's UAG, UGA, and UAA. It should be there somewhere. This is stop. Oh, these are the ones. Yeah. UAG, UGA, UAA. So, people, if those who are studying biochemistry, like I was studying biochemistry, I mean, these were obviously, I have to remember, I can't like look at the tables and such, like it was expected that I know, but I'm not expecting you to memorize this at all. All you need to do is use the table. Now, please, everyone pay attention to the question that was being asked. Very good question. Why is there a 5 in the beginning and 3 in the, what are the numbers doing here? Okay, so I'm going to address that by going back to our slide where I told you about numbering. No, not here. It's right. Oh, actually it's before this, right here. So if you see here the nomenclature of the sugar, you have 1, 2, 3, 4, 5. Okay. So if you see here, this strand is starting at 5. So carbon number 5, yes, that is correct. So 1, again, 2, 3, 4. And you may say the sentence, no, there is a carbon here. We are smart people. We know whenever there is a little corner, that's a carbon. We're not going to be fooled by the corner. That's a carbon, 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 carbon. So this is carbon number 5. And so that is the 5' end. So now you may say, what is the 3'? Look at that, where the DNA ends. This is where it ends. 1, 2, 3. Starts at 5, ends at 3. Starting at 5, ending at 3. That's why 5' to 3'. And look at this one. This is opposite. This is, you see the O hanging up. I mean, O facing up. O is facing down in here. So this is the 5, and that's the 3 there. You see? Same thing. 1, 2, 3, 4, 5, 5 prime. And that is 3 prime. And we'll show you one more time this same exact thing, because I created a slide specially to explain this. And I think we went over it last time, but I don't mind going over it again. If you see here, 5 prime, 1, 2, 3, 4, 5. So you have 5 prime here, and then it keeps going, and you're ending with 3. And here it starts with 3 and ends with 5. Look at the 5 prime and look at the C. 5 prime, C, AAT, AAT, CGT, that is CGT, and the final is CA. So do you see, and then it ends with the 3 prime. So you see how I actually went, I'm going to show you by using this thing. I actually went from here to here to here, and I'm counting it. You know, I'm just kind of going through it all the way. I'm following this, and that's exactly what is 5 prime to 3 prime. Oh, that's also another very good question. So someone has asked me, why is one facing up and the other facing down? Great question, guys. Very nice questions. And that is why, you know, it's always nice to be able to study a little more together. So I would say that I think this is happening because of these bonds. If you look at these hydrogen bonds here, AT forms 2 hydrogen bonds and this forms 3 hydrogen bonds. It is, if the orientation is different, these bonds will not be able to be formed. If it is 5 prime to 3 prime, how will we align this? Because then 5 prime, okay, let me just say it's going to look exactly like this. It's going to be like you have to, trying to think. Okay, give me one moment. Let me bring something that can help you understand this. Okay. So I have something for you. Let me stop sharing for a minute. So I hope you can see. So I have 2 combs in here, okay? When my 2 combs, I would actually ask you to switch to a speaker view. I don't know if you can see this. Sometimes you can, yeah. So if the combs are just, you know, they are both going in the same way, that is 5 prime to 3 prime. But that won't form a bond. So you actually have to invert one so that they can actually form the hydrogen bond. Unfortunately, don't have 2 red combs, but for them to form a bond, one of them have to be inverted. If both of them are facing the same direction, cannot form the bond. That's all I have to say here. So for you to form the bond, they have to be, one has to go from top to bottom, other has to go bottom to top in order to form the hydrogen bond. Yes, always. Good questions. You know, you guys are amazing. All these are great questions. Yeah, it's always 5 prime to 3 prime and always 3 prime to 5 prime. And that does not happen in RNA because RNA doesn't even have 2 strands. So for these hydrogen bonds to form, that is essential. Now, I don't want to take it in different direction, but I do want to tell you that this is so important. This is how all the organisms have evolved. You can take this really small microscopic bacteria, it has the exact same ATGC. You can take a banana, it has ATGC. You can take a ladybug or a tree or anything, everything has the same DNA, everything is double stranded, everything is 5 prime to 3 prime in one direction, 3 prime to 5 prime, and everything is the same. This mechanism stays the same. The only people that, not people, the only organism that violate this is virus, because they sometimes say, I don't care about DNA, I'm going to be an RNA. So that's the only where it is like, oops, sorry. I was probably talking without this thing. So I was saying is like that the only time this gets violated is when it is when it is a virus. So let me just show this one more time. And like I said, if you want, you can switch the view to the speaker view. And that is if the DNA, both of them are in the same direction, you won't be able to form a bond. You have to actually turn one around so that these can kind of align with each other. Okay. So for it to align, you have to form it. Otherwise, if both of them are switching the same direction, you won't be able to have the bond form. So that is why you must make sure that one of them is 5' to 3' and one of them is 3' to 5'. Sorry, I was speaking without the earphones, so I repeated some stuff. And like I said, it is a basic property of all living organisms. So this part, you have to make sure that you remember. I don't so much care about the history. History is great from understanding viewpoint. You also need to know this. You will be working in all these different, whether in nursing or in dental hygiene, you'll be constantly working with mostly a lot of you with, the human health. There are about 30,000 genes, and, the genes are extremely important in terms of what they control. And the genes are nothing but of a fixed sequence of DNA. It's a particular sequence of DNA that make up a gene. And then that DNA can be translated to RNA to protein. And I already discussed this with you, why your eye is different from your muscle cell. Everything has the exact same copy of DNA. However, only certain parts of those DNA, certain genes would be triggered to make RNA and RNA to protein. So eye cell is completely different from your muscle cell, even though they have the same DNA. Okay. And you know that.
Dr. F,
Yes, my
I'm
dear.
sorry. So I was just trying to confirm. So the 5 is always going to be in front of DNA, and then 3 is always going to be in front of the RNA. That's just always how it's going to be. It's always going to have a 5 right there, and it's always going to have a 3 right there.
Oh, this is just the DNA. This is not RNA. Okay, I see the confusion.
Okay.
This is just the complementary pair. I'm just showing you a complementary pair.
Okay.
So that's what it is. Now, if you really want to know this, I'm going to tell you, but I hope that this is not something that will confuse you. When the RNA is getting formed, remember, RNA doesn't have 2 strands. It has only 1 strand. So I'm going to give you here something here. This is called the template strand. I'm sorry. So the RNA, whenever it needs to be formed, it is formed from the template strand. So it's going to look exactly like the coding strand. I know this is going to confuse a lot of people. And this is not even, something that I don't want to confuse anyone, but I do want to tell you that the RNA is going to look just like the coding strand. The only thing is, if I was going to make RNA, if you say, how are you going to make RNA? This is what I would do. I would write here C, A, U, C, G, U, C. Will I write 5' to 3'? Yeah, you can write 5' to 3'. That is correct. So that's not a problem. Okay. So we don't have a whole lot of material left to study in terms of like me giving you hints on what all, but this is something important and you need to know this. And this is how your RNA is going to look like. It's going to look like the top strand, and just it's going to look a little bit different. Okay. Is there any questions at all about any of these things?
Yes, I'm sorry. I know that everybody always write in the chat, but I'm just gonna ask, for the last assignment, assignment 4, it was I believe it was the last question. It had like a bunch of questions on one graph or formula, I believe. Is there any way that you can go over those questions?
Yeah, I can definitely do that because I don't want you to feel like that is going to be something you don't know for exam. I can definitely do that. Okay. Let me just go ahead and just give you a few more tips, and that would be pretty much what you guys are doing. And for the rest of it is just like how it gets damaged. You can read about it. You can read about the direct and indirect way of. So the exam is 1 day. How is it 1 day, my friends? That is like the whole 1 week. The whole 1 week. You are going to finish the exam by Sunday. You have the entire week. The only thing is you have only one attempt. Study, take your own time, sit down and do it whenever you want to. I would never give a 1 day exam. That's like I'm sorry, yeah. So thank you for clarifying that for others if they were thinking. You can do the exam anytime next week. Okay. Yeah, I'll go over that. But I'm just saying you can definitely take the exam anytime next week.
Is it
And
timed?
It is timed, but there is enough time. Like usually the number of questions, like if it's 40, the exam would be 70 minutes, something like that. Because a lot of them are multiple choice, like 1 minute question. And the only reason it can take time is if you're like going back for every single question. That could be the only problem. So, I really, really want you to honestly do a great job. And trust me on this, guys. If in the final grade, if you are at 89.3, I will tell you this is what you can repeat and see if you can bring up your grade to an A. I don't think from my side, I would think that if you have an 89.5, you almost deserve a chance to be in the next grade level. Okay. So don't fear so much for the grade. When you do the exam, you do it with full honesty and integrity. And please tell me if something was hard or something that I can help you with. I would rather you approach me in that way than trying to do something which is not right. Okay. So let's try to stick to that. I'm not going to go over this. These are things that we have gone over. These are things you can read and learn about them. What are the different types of uses of radiation? And we do know that cancer therapy does involve, radiation. So with this, I'm going to stop sharing this part. And I do want to go to one of the I'm sorry, this is like some stupid CNN stuff open. Okay. So what I was going to tell you is I'm going to show you one more thing before we switch over. Okay, so I'm going to get you to one of the very important things which you would have, I bet, never seen. If you've seen, I'll be really happy. So this is the database where all the genes can be found and all the publications can be found. So for example, let us think about any gene. Okay, so I know everyone knows insulin. So I hope you can see my screen. If you can't, let me know. So I'm going to say insulin, and I'm going to mark it by saying, and Homo sapiens, just looking at human insulin, it will still come up with a lot of searches. Okay, so whenever I do that, if you see here, there are so many books available. There are so many catalogs, so many things. There's so much information, genes available, proteins available, all of these. You can see there is so much information that is available on insulin. I think we should have said insulin gene, and that probably would narrow down the search a little bit better. And if you see here, there is so much information that is available. I'm just going to take a minute and show you one of the genes. because I would like you to see a real gene, not like one of those fake genes. So look at this. This is how people who are studying these, how they would do it. So let us take, I don't see the, I want to do Homo sapiens. So I'm going to click on the, if you see on the right-hand side here, there is this mus musculus is the mouse. Homo sapiens is us. Rattus novergicus is the rat, and so on and so forth. I'm going to click on Daniel Rio, I think is the zebrafish. So there are all these different ones, and I'm just going to click on the Homo sapiens because I'm just interested in seeing about the human genes. So if you see here, this is this is about the insulin. And then I'm just going to click on this one, on this particular one. Okay, so just for the sake of showing to you, okay. So there is lots of information that is available. You may wonder, why am I showing this to you? I'm showing this to you to tell you that, you know, this is how people study some of these genes, okay? And just for your information that, I'm trying to see if I can pull up the code for it. Maybe the code is all the way down. Sometimes these things change a little bit. So source sequence, okay. I'm trying to go to the top one, source sequence, this, related this, okay. I think I clicked on the wrong one, but that's okay. We can always go to the right one. Give me 1 second. I'm just going to go up and oh, this is the FAQ. I'm sorry. I've been clicking on wrong things. Hold on. Okay, so I think that's where we are. Go to nucleotide and I'm going to the gene bank. I should have actually clicked this one. Okay, so this is, we are looking at human chromosome number 11. And if you see here, I'm not sure if it's going to, I think it did pull up the right, one of the right genes. If you see here, this is what an insulin, one of the precursor looks like. Do you recognize A, G, C, and T? ATGC? If you see here, this is what an actual insulin precursor would look like. You can clearly see it goes from 1 all the way up to at least 1400. Actually, it goes up to 1431 or something like that, if you see here. So this is how typically how big it is. And now I have to tell you that there is a very, you know, there are many websites which can help you in conversion of this gene to protein. You know how what you are doing one by one, converting A to T and G to C, that can easily be done with the help of, you can just pick up all of this and put it in a sequence, and it can help you translate to a protein. So just for your information, that this is how people study. When you talk about bioinformatics, when you talk about how this are done, so this is like a real part of one of the part of chromosome 11, which encodes for something specific. And if you put it in the right, it's just one region. You can see it's a very big region. And if you just put it in the right format, then it will give you the sequence of a protein. And that sequence of a protein will be all in terms of in terms of those the codons converted to amino acids. Okay. So just to give you a idea of a real gene, real protein, and how this actually works. I think I needed to tell you something, right? What did I say? I'll tell you, share with one more thing in the end. Someone asked me. Oh yeah, the assignment. Okay. Let's go to the assignment. I mean, in HHMI, there are a lot of wonderful movies where you actually see how people find genetic causes of different things. And that when they are referring to the gene sequence, when they're referring to chromosomes, that is what they are doing. I'm just going to take you to the assignment number 4. Assignment number 4 for this one. And if you have already done it, it's fine. If you have not done it, I'm not giving you the answer. However, I would like to give you enough hint so you feel very comfortable doing the assignment. And like I told you, if you finish it, I'm going to grade it because I do need to make sure that all the stuff is graded to go in as a midterm grade on Monday. So please do not delay. Please try to finish it so that I can put a good grade for you. I'm sorry, this is not what I wanted to do. I just want to go to the assignment 4. Okay. So I think one of you have asked a question about, is it about this one?
Yes, I did not understand those questions in the graph. Like, yeah, I hope I'm not the only one.
Okay, so if you did not, yeah, I'm this, I think I can ask you a lot of questions and that will help you. So if you remember, a nucleotide has 3 parts. So for me, the key part is the sugar because that pentagon structure will help me identify the sugar. So I expect you to be able to pick that 5-membered ring structure to say that that is the sugar. And then the square would be the base and the circle, because I know that there is a phosphate backbone. If you see here, this is the phosphate backbone of the DNA. So that's the, and phosphate is negatively charged. That is why DNA is negatively charged. So this is the phosphate backbone. And this is where the bases meet. And we talked about this. And do you see how this is opposite? This is facing up, and this probably didn't do this right. This should have been facing down. It should be reverse. It's in a way doing it, but not doing it just, oh, it probably is facing down. Yeah, just it's very equal in nature. So this is facing up and this is facing down. So I want you to circle one part which shows the 3 parts of a nucleotide. What are the 3 parts? We know the 3 parts are the sugar, the phosphate, and the nucleotide. So I would like you to do with a pencil and please show to me what is a nucleotide. Okay. And so what we are talking about here, let me see if that to everyone. Okay. So then about these bonds, I'm going to take you to an actual figure so that you can identify these bonds and you're not limited to your imagination in thinking, but actually go to one of the slides. Now about the understanding of base pairing rules, you can put here anything, but please make sure to put a complementary base here. If you put A, you know what to put here. And if you put a C, you know what to put here. So you can put anything. And remember, if you put a yellow as C or A, whatever, you should put a yellow as C or A here. So just reminding you that you are going to use complementary base pairing rules.
Wait, okay. So I'm so sorry, Dr. M.
No, go ahead.
You're talking about number 2 on this question?
I am talking about, so number 1, I already said, number 1 and 2, I talked in the first go, because number 1 and 2 is about what is a nucleotide. Okay.
Yes.
So once you have identified what is a nucleotide, you have to circle the 3 parts of a nucleotide. So once you have identified and labeled them, now you have to demonstrate the base pairing rules here, because this is where the base pair in the middle is the base pairing is being formed. So DNA is attached in 2 places. If you see here, the purpose of this question is to make you think DNA is attached here, DNA is attached here. That is why the DNA remains as a double helix. If there is no bond here, it's going to fall apart. What is this bond? What is this bond? But before that, we are answering, if I put an A here, what should be the complementary base here? So you can put an A here, but you must put the complementary. You can also put a G here, but make sure to put complementary base pair to G for this one.
Okay,
You
so
know
that would be
what is-
C. So
Yeah,
if I
like
put
that.
G there, it would be C. Okay, and just go all the way down for all the bases, the all base boxes.
Yes, yes,
Got
but
it.
put the same colors in the same thing. Don't put 80 here, 80 here. You know this should be the same color. So accordingly, you're smart enough to identify that the colors could mean the same base, right? So
Okay,
whatever you put A here, you must put A here, okay?
got it.
And then what are the three bases on one strand of DNA and RNA that are called codon? It's just asking you that code, called the code for an amino acid. So when three of the nucleotides that code for an amino acid, what is it called? And that definition is in one of your slides. So we are going to go back and look at that slide, okay? Now, about the last question, so someone asked me like, what are these bonds? So that is definitely a very good question. What happens is a lot of times when we are looking at the PowerPoint, we overlook some of the information. So the questions are meant to help you review that information. And so good, you are asking that. Let me take you to this part where we have this main structure. I have this structure so that we can, oops, I think I went too fast. Was it this one? I'm wondering, where is it covered? I thought it was covered in that. Ah, right here, right here. That is actually correct. That is actually correct, Kayla. So let us look at this one. So this bond between oxygen and hydrogen or nitrogen and hydrogen is mentioned here. It's a hydrogen bond. You must remember that adenine and thymine form 2 bonds and guanine and cytosine form 3 bonds. So what stabilizes the middle? What stabilizes this ladder in the middle is the hydrogen bond. Now the question is, what stabilizes these back or the backbone of DNA? What stabilizes that? Can you tell me?
Would it be
What bond
phosphate?
is? Yes, that is actually correct. Let me actually write this down. I know a lot of times when I ask question, it may look like I'm trying to trick you, but I'm actually trying to ask you questions which will help you think. Okay, so that's a phosphodiester bond. So if you remember the ester is something O. So let me just show you here by labeling it. This is one ester bond, and this is a second ester bond. So if you write phosphate bond, that is fine, but it's more appropriately a phosphodiester bond. Or you can, if you said bond with the phosphate group, that is fine. So this bond in the end, and see this negative charge. So the negative charge, it brings a negative charge. This is not any charge, just for your information. It brings a lot of negative charge. If you see that this is a negative charge, this is a negative charge, this is a negative. You can see here the DNA is completely filled with a negative charge on the side just because of this phosphodiester bond, because of the way it interacts with oxygen and makes. So this is a negative, DNA is completely negatively charged because of the phosphodiester bond. Hydrogen bond, of course, does not create any charge. So those are the things to remember when we are marking those bonds as to what they are. So this particular slide is very important for your understanding, for knowing how the mechanism of, the basis of the structure of DNA is. Like, what is the forces behind, what are the bonds behind making it actually together? And if you see here, these colors are also indicative of the kind of thing I was telling you. Adenine always pairs with thymine, or thymine always pairs with adenine. You can see green blue with a blue green. You don't see a blue with a yellow. If you see a blue with a yellow, what would you say? What is that? That is a mutation. Like I said, the mutations can happen but that's what is a mutation. So I want to stop sharing at this time and